this metabolic signature. So you can ask question in between. So don't be troubled to understand this. So I had this picture. Just give me a minute. Yes. So I wanted to show you this picture. This is quite clumsy, but you had to see. You need to see this to understand the complexity and variety of metabolites present in an organism. So these are various metabolic pathways which are forming a very complex network, and among all these, there will be many metabolites which can be used as signature or marker of a particular situation. For example, uh, if I'm correct, progesterone is one metabolite which is used as signature or marker for pregnancy. And this is only one metabolite. But many times there can be a set of marker metabolite which can be used to, to understand the physiology of an organism. And here you can see some metabolic pathway has been uh, elaborated. The one with which you must be familiar is the carbohydrate metabolism, and in that you must be at least familiar with the pyruvate pathway and the citrate cycle. Pyruvate pathway through which glucose converts into pyruvate, and pyruvate is metabolizes in the mitochondria and finally converts into carbon dioxide, releasing the energy. But this pathway which you are studying in your syllabus is not in isolation, rather it is in connection with so many other pathways. Okay, it is connected with so many other pathways. That means there are many enzymes which are working in collaboration to decide overall profile of the metabolite. Okay. Similarly, you are seeing this is energy metabolism, this is lipid metabolism. It is showing how different kind of lipids, how are they being formed here, and of course they are all they work in regulated manner. And you are seeing that this lipid metabolism is related to this pyruvate pathway. And here also you are seeing that biosynthesis of secondary metabolite is also related to this Krebs cycle somewhere. Similarly, here also energy metabolism is related to it. And in this way, all set of metabolic pathways are some or other way are interrelated. Okay. And of these uh, metabolites, which might be in the order of 10 to the power 4, of all these, there will be few metabolites which will be marker of a particular condition. One example I gave was progesterone, and similarly, in some cases, it cannot be one, rather more than metabolites will together will be marker for a particular physiological condition. So that's what is written to generate metabolic signature. So one aim of the metabolomics is to identify metabolic signatures. Could you see the slide? I hope the screen is visible to you. Can somebody tell? Can you see the slide? Why metabolomics is relevant? Anyone? So to generate metabolic signatures. So in case of pregnancy, you know about progesterone, but for many situations, you might not be knowing which molecule could be signature. For example, uh, take air pollution. If there is air pollution, some kind of pollutant suppose has occurred in the environment, and you want to know what kind could you be able to assess by studying the metabolic profile of the humans living there? Of course, you can. You might not be knowing those signature molecules, but there are ways in metabolomics to identify those signature molecules. In that way, you can find out the signatures. For example, let me tell you that in our lab, there was a researcher who was studying the effect of uh, air pollutants on uh, auto rickshaw drivers and traffic policemen. And he didn't know the molecular signature, but when he studied signature molecules, on the basis of which you can easily assess whether the person is affected with pollutant or not. Okay. So molecular signatures, metabolic signature can be identified by adopting at least one approach of metabolics. Another is monitor or measure the metabolite flux. Metabolite flux means how many molecules are being generated by a given particular pathway. And you saw that there are many metabolic pathways which are working all the time in our body. And you want to suppose you want to measure how the Krebs cycle is working or how the oxalate is being metabolized. You know, many times oxalate when deposited into the body, it forms the stones. And to understand whether some given person is prone to get those stones or not, then you might be interested in studying the oxalate flux in that person. And in that case, you can uh, use this metabolomics approach for that study. Next, monitor enzyme pathway kinetics. So similar approach you can study to study any enzymatic pathway or molecular pathway, whether it is working or not, or at what pace it is working, you can study. Monitor gene environment interaction. How a set of particular gene is giving the metabolite in given set of environmental condition, you can easily study. Suppose there is a gene for stress tolerance, maybe drought tolerance in the plant, and you want to check whether this gene is working properly in saline soil or not, and then you will grow that plant in different salinity level and slowly create the condition of drought and check whether those marker of drought are being expressed or not. So in that way, you can monitor the interaction of gene and environment. by studying a set of metabolites, those which are signature of that particular kind of trait. Track the effect of toxin, drugs, or surgery. Suppose you have taken some kind of toxin. Of course, after some time, you might not that toxin might not be detectable. You must have heard of that. Many uh, athletes used to take steroids for better performance uh, in their sports, though it is illegal. Suppose some a given sportsman has taken some kind of steroid, and after 1 or 2 hours, that steroid has been metabolized. You won't find the clue of that. But again, that steroid must have changed metabolomic profile of that sports person. And by studying that metabolic profile, you can find out whether that person has taken that steroid or not. Monitor the consequences of knockouts. Okay. Suppose you have knocked out some genes in the plant or animal, and you want to see what kind of result uh, you are getting there, you can easily find out. Identify the function of unknown gene. Many times you might encounter some metabolite for which you don't know the enzyme, which pathway it is adapting to come into existence. That means it is uh, coming from some genes which are yet unknown. So you can also go to find out those unknown genes. Okay. There are two related terms, metabonomics and metabolomics. Metabonomics is actually quantitative study. The quantitative measurement of time-related total metabolic response in vertebrates to pathophysiological, that is nutritional, xenobiotic, surgical, or toxic stimuli. Means you want to study the metabolites quantity in the organism. And what is metabolomics? The quantitative measurement of metabolic profile of model organism to characterize their phenotype or phenotypic response to genetic and nutritional deprivation. So metabolomics is a kind of theoretical study, and metabonomics is a kind of, you can say, diagnostic study or application of metabolomics, and both are quantitative. There are both some related terms. Metabolic profiling. We see this. The session for today is for the introduction of metabolomics to you, and from the next class we will see some techniques which are utilized in this particular field. So what is metabolic profiling? It is quantitative study of a group of metabolites, known or unknown, within or associated with a particular metabolic pathway. So it is group of metabolite, and we are studying the quantity of those metabolites present in the organism. Metabolic fingerprinting measures a subset of whole profile with little differentiation or quantitation of metabolites. See, actually, metabolic fingerprinting is the same. Studying the signature metabolites, not the complete profile. Profile means you are studying the whole metabolome. And when you are doing fingerprinting, you are taking a set of metabolites for some kind of identification purpose. Then there is target isotope based analysis. Here, what we do, we want to trace what kind of, especially it is used for pathway analysis. You must be knowing the Kelvin cycle, how it was discovered. Actually, Kelvin took radio labeled carbon dioxide, and he used the model organism chlorella. Chlorella is a unicellular algae which has its chloroplast. So when this chlorella received radio labeled carbon dioxide, it must be engaging it into photosynthesis, especially the dark reaction. What Calvin did, he killed the chlorella just after giving the carbon dioxide, radio labeled carbon dioxide, after maybe 1 minute. Then he performed the experiment again, and next time he killed that after 5 minutes. And next time he killed that after maybe after 7 minutes or 8 minutes or 10 minutes, and so on. And he tried to find out which molecule is radiolabeled. So the first molecule he found radiolabeled was 3-phosphoglycerate. That means the carbon dioxide which was labeled radiolabeled has turned into 3-phosphoglycerate. And after some times he found that that other molecule is radiolabeled, then other molecule is radiolabeled, and in that way he did isotope based stressing of metabolic pathway. So it is also a kind of study of metabolite, and it is called as target isotope based analysis. Okay, so in this approach focuses on particular segment of metabolome by analyzing only few selected metabolites comprising specific biochemical pathway. As I gave you the example, there he studied the pathway of dark reaction of photosynthesis. By applying a similar approach, Krebs cycle was also discovered. And this is still being used for many kind of uh, for tracing down many kind of metabolic pathway as well as the pathways, molecular pathway through which protein is conveying the message. In both the cases, this target-based approach is being used. So this we have discussed. Metabolic food printing refers to the fingerprinting of exometabolite. See, there is another term, exometabolite, means metabolites which are secreted out by the organism. If you are studying those, then that profile will be called as metabolic food print, and the procedure will be called as metabolic food printing. So what is the difference between metabolomics and traditional clinical chemistry? Just I mentioned that in traditional clinical chemistry, we used to focus on one metabolite for diagnostic and other purposes. But here we are going to study many metabolites, and at the same time, simultaneously, we will study many metabolites for various purposes, clinical or non-clinical purposes. So more metabolites, greater accuracy, and higher speed. All three aspects are involved in metabolites. Okay. More metabolites you can study at a higher speed means within less time you will be able to study many metabolites and with greater accuracy than previously. Actually, because of these modern techniques and machines only, this and traditional clinical chemistry has turned out into metabolites. Here you can measure multitudes, tens to hundreds of metabolites once in one time with no separation. Separation means, usually previously for study of any metabolite, first you have to isolate or separate it out from other metabolites. Suppose you want to study a polyphenol from green tea and you want to quantify or identify it, then previously you had to go for maybe column chromatography or thin layer chromatography. Then you would use HPLC, and after that you will go for identification technique. Many times you might be required to go for a cleanup procedure and two or more stages of uh, column chromatography, and thereafter only you will be able to identify that one. But here, even without separating, just take the crude extract or the body fluid and put that into machine, maybe um, LCMS, and you will get the result. You will get the profile, and it can quantify also simultaneously along with the identification. It allows metabolic profile or fingerprints to be generated. I just mentioned that metabolic profile or fingerprint can be created for an unknown stimulus. So that is done. Mostly automated. Most of the machine nowadays are automated, so less effort is required for metabolic analysis. Little sample preparation and derivatization. Sample preparation and derivatization is we will discuss later how the sample is prepared. What is sample preparation? Whenever you will take the body fluid or any extract from any organism, we cannot directly apply that into the machine like NMR or mass spectrometry. So between that, there is a procedure called as sample preparation, and many times it includes derivatization also. But nowadays, these modern machine has reduced the cumbersome process of sample preparation, and it can be quantitative also, means you can measure the quantity, analysis and result in less than 60 seconds. So it is not that much feasible even today. Getting the result in 60 seconds is feasible only in very few cases. It is not that much easier. So, so at this level, it will be difficult for you to understand this. But I want to point out few things here that in this you are seeing that these are different metabolites like hippurate, fumarate, urea, allantoin, creatinine, water, hippurate. Here you are this is actually HPLC of a sample. It is showing that these are different metabolites present in that sample, and the area covered in this P is depicting the quantity of that molecule. So this is one approach where you are quantifying few molecules. Okay, but on the other hand, you can get the chemometric pattern also. Of course, it involves bioinformatics and procedure and statistical procedure. But here you can identify that. Suppose this is the control. Control means suppose there is a person which has got some metabolites which are similar to each other, but when they were applied with some drugs, some new molecules or new metabolites have been identified, and these can be the signature for these kind of treatment. Suppose a person has been treated with this ANIT, full form is okay. Suppose this is one drug, if this is another drug, and when the person was not given any drug, these kind of metabolites were found. These metabolites are actually similar to each other, that's why they are placed together. And these new molecules were identified when those persons were exposed with this drug, and when they were exposed with this ANIT, new kind of metabolites were found in that person's metabolite. In this way, a chemometric pattern of particular treatment or a mental condition. quantitative. Okay, so this is the simplified uh, procedure in metabolomics approach. How are going to study the metabolic profile of a sample? So here is the biological sample. This is a plant, or it can be meat or some other animal tissue. Suppose you want to study the metabolomic profile of these organisms. First, you will have to take the sample out, put that in some vessel where you will rupture the tissue. This is the device, ultrasonic device, which will create ultrasonic wave here, and that will rupture the tissue. And after that rupture, the small molecule will come out into this liquid present in this vessel. And from there, you will filter it out, other things, except the metabolite you want to study. And then there is a sample procedure, sample preparation procedure. Why? You know, we have different kinds of metabolites in a particular organism. It can be sugar, means the class of carbohydrate, or class of lipids, or class of phenolics. There are different kind of metabolites present in the organism. Suppose you want to study only lipids. So you, before putting that into the machine, you will try to clear the clutter. Clear the clutter. You will want to remove all the metabolite except the lipid if you want to do so. So that is done in the sample preparation procedure. And after that, you will apply that to the machines, for example, HPLC or gas chromatography, or NMR, or nowadays we have ESR, or nowadays spectro is directly applied in many cases. So these different machines will work to give you the profile of metabolite. So these are the steps. One is the extraction and the sample preparation. Many times both are considered as one step of sample preparation. And then running that into machine to get the metabolic profile, and thereafter you will do the bioinformatics part or statistical analysis part. And considering that, when you will go through this process, two main approaches of metabolomics has been classified. One is untargeted, another is targeted. As the name suggests, untargeted is meant for discovery. Suppose you don't know the metabolomic profile uh, of, suppose, human in certain condition, maybe pollution. You don't know that. Suppose there is a gas leak has happened in a particular area. Gas leak from some chemical industry, and humans in that area were never exposed to that gas. That gas is toxic. They were never exposed to that. Now you don't know what kind of metabolic profile would be there. You don't know what kind of molecules going to change in the humans were exposed to that gas. So in that situation, you will want to study the whole metabolic profile, and you would want to find out which can be which metabolites or molecules would be marker or signature molecules. So here, hypothesis is not created in the beginning. You don't have any presumptions before the study. Okay. Just you will take the sample, you will try to get as maximum as metabolites into the sample, and you want to study it. So it is kind of global and comprehensive analysis. They don't have any target that we want to study this pathway or this set of metabolite or this particular metabolite. When you don't have any target, then it is untargeted study. It is global or comprehensive analysis. MSMS analysis, mass spectrometry, mass spectrometry related and database libraries are used. अब ये कैसे काम करता है? We'll see that later in next classes. So I was talking about targeted and untargeted metabolites. So here in untargeted one, we go for qualitative identification. And quantification is difficult. We don't go for quantification, though relative quantification can be done. And how it is done, so here there is, suppose you know molecules of the pollution gas. Suppose a gas, this kind of gas, and Sorry, participants. There was network issue on my side. So, yes. So I was giving example of targeted metabolomics. That you are having some hypothesis in hand, and on the basis of that, you want to study only selected metabolite for some diagnostic purpose, maybe, and that will be called as targeted metabolomics. It is done for validation, not for discovery. And here, quantitative analysis can be done. Here, the molecules you are going to study is already known. You know that how are you going to identify, and therefore you can do the quantification also, absolute quantification. Actually, what is relative quantification and absolute quantification? You will study that in detail when you will study SPLC or GC. But for right now, I can tell you by this graph of this data analysis, you are seeing that this is peak 6, this is peak not okay, suppose this peak is broader, as compared to this peak. So certainly you can see that the metabolite which belongs to this particular peak is having higher quantity than this one, because the peak area, actually the area covered in this particular peak depicts the quantity. Okay. So here relative quantity can be known. You can say that this particular peak is having concentration higher than this one, and in fact you can also quantify how many fold it has higher quantity in the sample than this one. Okay. But what is absolute measurement of quantity? For absolute measurement, you will run, or you will get the same profile uh, from the known concentration of selected metabolites which you want to study. When you will run a known concentration, means set of metabolite with known concentration, you will know that this particular area belongs to this much quantity. And therefore, after that, correlating the area of the two profile, one of the unknown sample and the from the known sample, you can find out the, the absolute quantity. Okay. So that's what is written here. When you are doing the discovery, you are not knowing the compound. So how can you prepare the known concentration? So in that case, you will do the relative quantification, but you will do absolute quantitation when the that metabolite is known and that is available with you. Okay, when it is available with you, then you will prepare solution of known concentration, and that solution you will run as a sample. You will get the peak area, and you will correlate the peak area to get the absolute quantity. So there are two approaches: untargeted and targeted. So untargeted is used in discovery, while targeted one is used when we want to uh, do some kind of diagnostics. So this is targeted and untargeted. This is again depicting the uh, elaborated path, or you can say the main steps involved in targeted and untargeted metabolomics. So here you are seeing that the question is, what are the level of specific metabolite in sample? Okay. For targeted one, you know which metabolite you want to study. Okay. And you want to quantify those. What is the level? Means what is the quantity of that? In that case, you will have standard metabolite. Standard metabolite means the known one. Suppose you want to study the level of fumarate in the plant under salt stress. Fumarate, it is a metabolite of Krebs cycle. You want to know, and you can easily buy fumarate from the market. So you will prepare a solution of fumarate of known concentration. Suppose 1 micromolar, 10 micromolar, 5 micromolar, different concentration of fumarate you have prepared. That will be your standard metabolite solution. and you will apply that solution for in some machine to study that. One machine is LCMS. Okay. What LCMS will do? It will create the peak. It will give you the result, and after that you will get the sample uh, from different kind of tissue where you want to study the level of humidity. Now, here the cells and blood are written, but in case of humidity, you can take some plant tissue. Okay. Just take the plant tissue again, do the extraction procedure, and run that in LCMS and get the similar result. Now compare the level of fumarate you found or the area of the peak you found from the known sample of fumarate here and the area of the peak in unknown sample of the fumarate, maybe that means from plant. And in that way you can quantify the level of fumarate. And here for untargeted, it is comparatively similar to perform, difficult to analyze. You will take the tissue and you will throw them in the LCMS. Here you are finding one peak or one metabolite. Here you will find thousands of peaks. So when you will get so many metabolites, it will be difficult to analyze it, and therefore you will go through computational analysis of these metabolites to find out to identify the molecules and to find out the metabolite profile which is being altered from one situation to जैसे आप समर्स में या आप सिंपल कहो कि आप सुबह आप अपनी मेटाबॉलिक प्रोफाइल आप चेक करना चाहते हो टेक सम ब्लड फ्रॉम इन द मॉर्निंग फ्रॉम योर बॉडी do this kind of analysis throw that in LCMS you will get these profiles and after having the breakfast or lunch again take a small blood sample and throw that into LCMS you will again get this kind of profile but certainly there will be some change though you won't be able to know what peaks are these but change you can identify in the two different situations. Okay. Before breakfast and after breakfast, this though you are not able to identify what these metabolites are, but certainly you will be able to find out the change which has been happened in in these peaks, and therefore you will identify which peaks are actually changing after having the breakfast, and those peaks means those metabolites which belongs to those unique peaks which are changing after breakfast can be the signature molecule depicting what is happening within you after having the breakfast. Okay. So these are the key stages of metabolomic study. Design the experiment. If you are doing targeted study, how we are going to perform good series of experiment, how we are going to prepare the standard, how we are going to prepare the sample, how we are going to run sample into the machine, which machine you are using, LCMS, GCMS or NMR or some other technique. Okay, all this will be in the part of design the experiment. Then you will do the sample preparation. Means you will take the sample, take the tissue, then do the extraction procedure, then do some extra step of sample preparation. Then you do the chromatography for separation of metabolites. In many cases, you don't need the separation. With whatever the case, whether you're separating or not, after that you will go to identify the metabolite. And identification is done by many methods, but most popular one is mass spectrometry. So through mass spectrum, you will be able to identify, means you will be able to get these peaks. After getting these peaks, you have to analyze that. Okay. These peaks will come as a part of data acquisition, and after that, you will do the data processing. After doing the data processing, you will get some graphs like this. Okay, and what this graph is depicting actually, that will come as a part of analysis and interpretation. So this is general flow chart. How are you? These are depicting the key stages of metabolomic study. So here that has been given in elaborated manner. So steps are formulation of biological problem to be addressed, what kind of biological problem you want to address through your study or experiment, and you will decide whether you want to go for metabolism approach. If yes, then go for it. Then you will decide whether it will be targeted or untargeted one, and then you will decide what kind of sample you are going to take. It is blood or some other tissue or some maybe urine. In case of plant, it can be some kind of secretion. It can be some root tissue or maybe some infected area from a disease or leaf or or the axillary buds. Whatever the tissue you decide according to your study. Now sample size. What is the amount of sample you are going to take? That you have to decide. Then experimental condition to which sample is will be submitted. Submitted means in the way it will be given to run in the machine. That is whether you will carry the sample at 4 degrees Celsius or 0 degrees Celsius or at room temperature, that would decide. What would be the pH of sample uh, on which you will carry it. Frequency of sample collection. How many times and how often you are going to collect that. Metabolic quenching to interrupt the enzymatic activity. You will have to quench. I give you example of chlorella for the study of Kelvin cycle. I told you that after giving the carbon dioxide, he killed that chlorella. Why? Because enzymes are always active. They can convert one metabolite into another one. So therefore, when you were at whatever point you have decided to take the sample out, you must quench that. You must stop all the metabolic activity there. So that is done by reducing the temperature at -degree Celsius, or sometimes you can add some chemical to stop the metabolic activity, stop the enzymatic activity within the organism. Next, analytical platform to be applied. Okay. What kind of machine you are going to use? LCMS, GCMS, NMR, etc. Sample preparation studies must be defined at this point. What kind of sample preparation you will use? For LCMS, sample preparation procedure might vary from GCMS and NMR and so on. So for each technique, sample preparation is little different. Sample collection, easy, just take out the tissue, whatever you want. Sample preparation for targeted metabolomics, extract the complete metabolite. Not for untargeted. You will take all the metabolite. For targeted one, you will have to do the extraction procedure thoroughly. Just say, for example, I told you that suppose you want to study the lipids only into the blood. So what you will do, you will do the extraction procedure, and after that you will do some, you will play with some kind of solvent so that only lipid can be taken out and rest molecule are left. So that step is called as cleanup step. Means clean out the desired metabolite from the rest one. And for untargeted one, take out the sample, whatever you want, extract out, filter it, and just after that, you don't need the cleanup step. You can go for direct analytical tool like SPLC, GCM, etc. So after running that in SPLC, GC, and CE, what would be the detection technique? Actually, HPLC, UP, GC, CE, MALDI, these are actually separation technique. And after separation means suppose you have 1,000 metabolites, you can't study all of them together. So what you will do, first you will separate. Machine used to do this part actually. Machine like SPLC, GC, or capital electrophoresis. So after separation, you need to identify that. And there are many identification methods. Some are based on absorption, some are based on fluorescence, some are based on some emittance, and there is one most popular one technique is MS, which measures the mass of molecule, because almost all the molecules have different masses. So identification or detection. Next is statistical analysis. So many statistical analysis are there. Most popular one are principal component analysis. How it is done, that will be discussed later when we study in these classes. After doing that, you need to identify. Suppose you have got the mass spectrometry pattern, and that mass spectrometry pattern, or whatever the machine you are using. Suppose you are using the absorbance, you must have used spectrophotometer. Okay, and absorbance is giving some. Suppose you have measured it at some nanometer of incidence light, and you are getting this much absorbance. So machine will give you this kind of data, absorbance or fluorescence or mass. But identification based on absorbance or mass or fluorescence is altogether different procedure. So based on mass, these are different libraries of metabolites from which you can identify the metabolite. Suppose you have got a metabolite which has mass of 149.6 Dalton. So which molecule it can be? So you will go to these libraries, and here you will try to match that mass of that molecule, and it will help you to identify what kind of molecule it can be. And there are other softwares which will help you to trust the metabolic pathway. Suppose you have found out a set of metabolite and you don't know that which metabolic pathway it is indicating of. That Sorry, I'm facing so much network issue today. So coming to application directly, what are applications? So mostly application you will see here is in the field of animal sciences, mostly related to human. Other application related to plant sciences we will study in classes. So what could be the applications? There can be many. Increasingly, it is increasingly being used in a variety of health applications, including pharmacology, preclinical drug trials. Preclinical drug trials means you can use that into other animals. Before using any medicine on human for trial, you are using that on some non-human animal that is that is called as preclinical trial. Toxicology, studying the effect of toxins. Transplant monitoring. Suppose you have transplant some organ into some person and you want to monitor whether all okay or not. So you can use this metabolomic approach. Newborn screening. Okay. When there is a newborn, you can assess the health status of that uh, newborn. In clinical chemistry, it has lots of scope. Tools for functional genomics. It can be used to study the functional part of genomics, which means what kind of genome is there and what kind of metabolites are there in given set of conditions. And however, a key limitation to metabolomics is that the human metabolome is not at all well characterized. We, um, we haven't understood well even the human metabolome. About other. But still, it has application, it has utility for certain purposes. These are again different applications: toxicology testing, clinical trial testing, fermentation monitoring, food and beverage taste, nutraceutical analysis, drug phenotyping, water quality testing, organ transplantation, genetic disease tests, nutritional analysis, clinical blood analysis, clinical urinalysis, cholesterol testing, drug compliance, dialysis monitoring, MRs and fMRI. There are many applications wherever the metabolite can be used for assessing some kind of change in metabolic profile. Importance of metabolomics: it is non-invasive nature. Non-invasive means you don't have to cut through organism to do some kind of diagnostic purpose. Just see, even this urine metabolome is the best one. Why? Because it is using non-invasive technology. Or many times nowadays people are using are doing the breath analysis. But the breath you are exhaling out is having many metabolites. You know? Your breath has certain kind of order, and each person may have different order. Not only this, even the same person, and that order is because of some chemicals, volatile chemical present in that. And that volatile chemical can be identifier of that person. And not only this, the metabolomic profile of the exhaling breath, the air you're exhaling, can be different in different situation, and that can be identified. So you don't need to push some syringe into someone's body. In some cases, I don't know for which disease, um, it is required to take the fluid out of from your spinal cord, which is very dangerous. So this metabolomic profile, when it will be quite developed, even the breath analysis can be diagnostics for many diseases. So it gives a mean of non-invasive technique for studying uh, in the field of pharmaceutical, preventing the healthcare, agriculture industry, and so on. Biomarkers discovery and drug safety. Biomarker discovery and drug safety screen are two examples where metabolomic has already enabled the informed decision making. In the future, with the availability of personalized metabolomics, we will be potentially be able to track the trends of our own metabolome for personalized drug and improve treatment strategy. Personalized, nowadays we are having concept of personalized medicine. That means each person has different physiology, so the same medicine cannot be prescribed for it. And for that, we need to know, of course, the genome of each person. Along with that, metabolite, because the same person can vary in terms of its metabolic profile from one situation to another. Accordingly, the drugs should also be used. They benefit from the metabolomics in various levels, from product to stress testing in food industries, control of pesticide and identification of potentially harmful bacterial strain, to research in agriculture, crop protection and engineering, medical diagnostics and healthcare, and future application in like personalized medicine resulting in personalized treatment strategy. In the field of agriculture, the development of new pesticides is critical to meet the growing demands of farming. Metabolomics enables us to improve genetically modified plants and helps us to estimate the associated risks by allowing us to get a glimpse of their complex biochemistry via informative snapshots acquired at different time points during planting. Suppose you have changed a plant genetically via a biotechnological approach. What kind of metabolite it will produce, you can assess through metabolomic approach at various stages of its life cycle. And this may give a good signature of, what kind of health effect it is going to impart on humans which are going to eat it. Plant metabolomics is particularly interesting because of range of function of primary and secondary metabolites in plants. About 300 distinct metabolites could be routinely identified per sample a decade ago. And number is gradually increasing over time. So a decade ago, we could identify 300 distinct metabolites on routine basis. And nowadays, we are able to identify even more. And it will keep increasing. Next is biomarker discovery. I have explained it. Okay. Once you have identified the biomarker for any disease, maybe in breath or saliva or blood or urine, okay, and then you can do it. Nowadays, people are saying that even you can identify whether a person is prone to get a cancer or not by just studying the metabolomic profile of blood. So you can predict the risk of cancer by studying the metabolic profile of blood. You don't need to go for X-ray or other techniques. You just give your blood sample and there will be some marker metabolite present in the blood through which kind of cancer, stage of cancer, or some other disease can be identified. So once the biomarker for a disease is discovered, you can easily do the diagnostic of various disease. Let me tell you that there is a scientist in ICGB New Delhi. He was working on electric nose. He termed it electric nose. Actually, it was a device which was actually collecting the sample from breath. It was nothing but an absorbent. When you will place that absorbent while you are exhaling out, it will absorb the metabolite in your breath, and that metabolite, when it will be studied through gas chromatography, it can be used to diagnose the disease. Personalized medicine, I have told you that once the metabolites are known of a person living in a particular situation, personalized medicine can be given. You will know that whether this medicine will suit that person or not. You must have heard that this medicine suits this person but doesn't suit another person. So how that person is different from the another one? Of course, if we'll take the terminal point or end point of difference, then it is metabolomic. Means difference at metabolomics level. Our metabolite profile is different of one person than another. So that's all from the lecture for today. This was about introduction, though it is a quite vast topic, but I tried to give you a glimpse of what is metabolomics, what is studied in this particular branch, why it is important, and what is the general procedure through which you can go to study the metabolic profile, and what are its application. If you have any question, you can put that in chat box. Maybe I got disconnected. That's why I'm seeing only one question. Bhupen is asking, sir, can we 100% rely on result of GCMS? That is compounds predicted based on its library. No, you cannot rely completely, especially when you are doing the discovery part. Suppose you are identifying some marker through untargeted approach. Of course, you can identify that these are metabolites which are being changed in a particular situation, and you might consider them as biomarker. But you will have to validate through statistically, means you will do the same study with different people giving the same kind of treatment, and you will see whether it is giving the same kind of biomarker or not. And after that, suppose you have identified that that suppose you have identified in a person on the basis of metabolomics that this person has the chance of having lung cancer. Okay? But you won't get restricted there. You will go for some other techniques also. You know that whenever you go to the doctor used to prescribe more than one test so that he can be assured that the result he's getting is reliable. And if you are asking about the library, of course you cannot rely completely on the library. But first you clue without putting much effect you will get from the library. And what we wish to do, suppose you have identified a metabolite through mass spectrometry and having a library, then again after identifying that, you might suppose you have identified a metabolite from a plant as fumarate through the library. Okay, then to confirm that further, what you can do, buy the fumarate from the market, prepare the standard solution, and run it in LCMS along with the sample. Suppose in one case you have gone through the GCMS with the sample only, and in the second time you mixed that sample with the standard fumarate, then it should increase the peak of fumarate which has been identified through laboratory. So, what I can do, I can go to the picture to make it more clear. Yes. You are seeing that suppose this peak had been identified through the library that it is fumarate. Okay. But to confirm that, what you will do, you will run fumarate alone. First, the peak of fumarate should come at the same place where it was in unknown sample. Second, when you will add fumarate to this sample only, the size, the peak area of this particular peak should increase. And apart from MS, you can go for NMR also to confirm it further. If 2 or 3 techniques are confirming that it is fumarate only, then you can be completely assured. So not only in case of GCMS, but when you will do the research and you are doing and you are giving some kind of result, always you should go for more than one technique to confirm one result. So you cannot rely 100% on any result, not only GCMS. Is there any more question in the chat box? which I'm not able to see. Can anyone unmute? Maybe host or co-host. Geeta, can you unmute yourself? Okay, there is another question. Um, Saran, sir, shall we also study metabolites created by drug or only endometabolites? Yes, we can study. Actually, it depends upon the purpose. Suppose you want to see what is the effect of that drug. So, of course, drug might remain for a very short time in your body, and of course, drug doesn't remain as such when it goes into your body. It also get used to get changed because of action of some enzyme with that drug. But apart from that, under the influence of that drug, other metabolites will get changed into your body. Not only into your body, of course, in your in the metabolites which are coming out of your body, maybe in sweat or urine or the breath. So depending upon the purpose, you can study endometabolite or exometabolite, which are coming out of your body, depending upon what you want to study. Participants, you can write in the chat box if you're not able to unmute. Geeta, can you allow participants to unmute? Participants, you can write your question in the chat box. From the next day, we will study SPLC and GC. And if time allows, then we will study a little bit NMR also. Saran is writing that determination by MS is also common in proteomics. In what way it differs from proteomics and metabolomics? See, when it is used in proteomics, machine is set or programmed to 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 measure the mass of high weight molecule. You know, and when it is made for metabolomics purpose, then it used to screen only small molecule. It will not take uh, molecules of higher molecular weight, more than 1000 Dalton in fact. So So that is how it works. In fact, actually, the same machine can be used for both proteomics as well as metabolics. The same mass spectrometer. This mass spectrometer is attached with HPLC also. Mass spectrometer can be attached with GC also. But the two spectrometer used to be little different. One is made to detect out the molecules of higher molecular weight. Another is designed to to, to identify and quantify, not quantify, identify the molecules of lower molecular weight. I think you could get something. It is completely new branch. Even this is not in our syllabus. So, purpose is to introduce with this topic to you, so that if some of you can get interested, you can choose this field for your further studies. Welcome. If you have any other question, you can put that into chat box. Okay, if not so, I would ask if there is no more question, then I think you can leave now. I would ask the host to end the meeting. Geeta. Yes, yes, you can leave now.
Okay, sir. With this, can we just leave the meeting?
Yes, I was calling Geeta to end the meeting.
Yes, she's not
So,
responding. I don't know why.
yes, yes. So then ask her to-
Geeta, can you please end the meeting for all?